scientists, so we're excited to hear from them. Okay, any announcements? Okay, I'm happy to introduce today's speaker, Gideon Bradburd, who's a new postdoc in the MVZ and is working jointly between Bree Rosenblum and me, and he's spending 2 days a week over with you and 2 days a week here, and we don't know what he's doing on Friday. Gideon got his undergraduate degree from Yale in 2008, and then did his PhD at Davis, working with Graham Coop. And he says here, the questions that get me out of bed in the morning are, how are patterns of genetic variation partitioned across space and through time? And this reminds me of a graduate student I interviewed once, who's a prospective graduate student who wanted to work with me, and he's very earnest. I won't say who it is because you would all know him and it would embarrass him. But I said to him after some small talk, the first time I met him, I said, well, so tell me something. I said something very open-ended. So tell me something about yourself. And he said, well, I guess you could say what gets me out of bed in the morning is population genetics. And at first I thought he was just giving me a line because, he wanted to get accepted. And then I looked him in the eye and I realized he was telling the truth. So Gideon's the same way. That's what gets him out of bed in the morning. And well, there's some other fun facts here, but I've embarrassed him enough, so I won't take any more time. Thanks, Gideon.
Thank you, Michael, and thanks everyone for coming. I'm going to be talking about a few projects from my PhD and that are continuing, and then if there's time, I'm going to talk about some of the projects I'm planning on doing with Bree and Michael during this year. And I also just want to give a quick warning that there will be some conversation about non-vertebrates in this talk. What? So be forewarned. Okay, so thinking about variation as something I find really exciting, variation exists everywhere in nature. These are some of my favorite examples from California's herpetofauna. I probably don't have to talk about them to the room. In the interest of time, I won't. But suffice to say that there's a lot of variation in natural populations, and very often it's spatially partitioned. And understanding this variation is really important for understanding not only the process of speciation, but also the process of local adaptation, both of which I think are fundamental to our understanding of both ecology and evolutionary biology. So when we think about how variation is generated and maintained, we often think about it in a context of understanding how population differentiation happens. And the model that I like to think about is that there was some ancestral population at some point in the past, and then that population split, and once the populations or the subpopulations have become separated, they sort of change in flavor, which is represented by these changing colors, through time until they're sort of differentiated or differentiable in the present. So a quick cartoon of that is just is what I'm going to present here. So here are 5 diploid individuals, each of which is heterozygote at a single locus, and the 2 alleles are green and yellow. And if we just plot sort of random drift where each individual in each generation is choosing 2 parents in the previous generation, and that sampling is random, you can see that the frequency of these alleles is kind of bopping around randomly through time solely due to this random sampling process. And then if we introduce a barrier between these 2 populations, which is this gray line here, you can see that once they're not choosing parents from the other population in previous generations, once there's this barrier, they can start to diverge. And we can just visualize that really easily by plotting the frequency of the green allele over generations. And you can see that while there's sort of a single trunk population, the allele frequency bops around but stays kind of stable. And then once they've diverged, the allele frequencies are able to diverge. This is just a cartoon. It doesn't necessarily happen, but it can happen once there's no gene flow. But obviously, if there were migration sort of from one to the other of these 2 subpopulations, this differentiation could disappear, just become homogenized. And this is sort of how I think about population differentiation, is that drift is acting locally in opposition with migration to produce differentiation. And one of the main determinants, I think, as we've all thought about, of migration and differentiation is space. It's just the spatial structure of populations, because individuals tend to disperse and mate locally. And this sets up sort of one of the classic patterns in population genetics, which is isolation by distance. And it's summed up really nicely by this guy Waldo Tobler in the first law of geography, which is everything is related to everything else but near things are more related than distant things. And basically, it's just the action of migration in opposition to drift or selection happening locally that leads to this type of pattern of differentiation with distance. So here's just a cartoon of this. This is real data from human populations sampled globally, and you can see that at just this single locus, there's each of these pie charts represents the frequency of the different alleles in space. And there's strong spatial autocorrelation of the different allele frequencies, right? So this is, at a single locus, a pattern of isolation by distance being acted out in humans. But it also scales up to genome-wide data. So this is a canonical figure from John November's paper in 2008, where this is a PCA plot of humans sampled throughout Europe very densely. And you can see that this PCA plot really beautifully recaptures the map of the sampling of these humans. And I found out recently that John had to do some jiggery-pokery to get it to look this nice. But the basic pattern is very real, and it's this whole genome pattern of differentiation recapturing the spatial pattern of the sampling. So there's a lot of different processes that shape patterns of genetic variation. And those include dispersal limitation, demography, bias dispersal, local adaptation, environmental heterogeneity. There's lots and lots of them. I'm not going to talk about them all, but I'm really interested generally in figuring out what are the patterns of population differentiation, genetic variation, and what can we learn about the processes that have acted through time and across space to shape those patterns. So today I'm going to talk about two projects, one from my PhD, one that's ongoing, and then one that's sort of future directions with Bree and Michael. And I'm going to start by talking about a project I've done looking at population structure and admixture. So these are data from human samples. This is 95 human samples taken from across the globe. And what I'm plotting is allele frequency covariance against just great circle geographic distance between the samples. And you can see that there's a lot of noise, but there's also a very clear decay of this covariance or this relatedness with geographic distance. And we can zoom in on this by coloring each point by the pair of samples it's comparison between. So this looks a little bit like a laser light show, but I really like it as a way to visualize data. So if it's a comparison between a pair of samples is colored on the outer margin by one sample and the inner circle by the other sample. And when we do that to this data, you can see some patterns immediately pop out. So one is these comparisons with red or fuchsia inners and purple outers are comparisons between East and West Eurasia. And you can see that per unit geographic separation, they have less covariance, they're more differentiated than samples that are comparisons between Western Eurasians or East Asians. And this is potentially a really good indication of barriers having acted in the past or barriers acting in the present between gene flow crossing Eurasia. And one obvious hypothesis would that be this is the Himalayas acting as a huge barrier to dispersal. Another thing that pops out is these bottlenecks. So these are all comparisons, these red and yellow comparisons are between samples from the Americas. And you can see that, again, per unit separation, they have very high covariance with each other. And this is potentially reflecting a shared history of bottlenecks, giving them a lot of shared drift following the expansion over Beringia into the Americas in the recent past. So that is a nice way to visualize data, but we also, sort of as the first pass in all population genetics studies, we want to know what do we have? What is the structure in the data? Who is related to whom and how? And there's a lot of methods for doing that. So a really common one is structure. And I'm going to come back to talk about structure in the second part of the talk. But suffice to say for now that structure infers, as you'd expect, discrete structure. And a lot of times when you have a pattern of isolation by distance, although structure is very powerful and informative, it's also in some ways an inappropriate model for continuous differentiation. Another model that's recently come out is TreeMix from Jonathan Pickrell and Joe Pickrell and Jonathan Pritchard. So this infers a population phylogeny between samples and then draws these admixture arrows onto that phylogeny. TreeMix is really cool, it's also really powerful, but again, when there are these spatial patterns of differentiation, a lot of times this hierarchically organized structure is not the appropriate model to describe that. And then lastly, I think one of the most widely used models now is principal components analysis. And there's a lot of extensions that have happened recently around this type of model. Principal components analysis has no sort of explicit population genetic model, which makes it very flexible. But it also has sort of some problems with its interpretation, and I'll highlight those problems as we go on, but there's a really great paper about it from John Novembre and Matthew Stevens in 2008 that I'll just be paraphrasing, so I encourage you to check it out if you have further questions. So we wanted to try to come up with a method that explicitly models spatial patterns of genetic variation and does so in a way that avoids some of the pitfalls of PCA and its interpretability. And we're calling this SpaceMix. SpaceMix uses genetic data with a very simple model of isolation by distance to make what we call a geogenetic map, which describes genetic relatedness or similarity with distance on this map, or proximity. And as of like this week, you can get this as an R package now, just off of my GitHub. And I guess this will be taped, but also it's on my website, and I'm happy to talk to anyone after this if you have any questions about using it. So basically the idea with SpaceMix is that we use sample covariance in allele frequencies across loci, which is the extent to which a pair of samples co-deviates from a mean allele frequency at each locus. And we model that as a parametric function of geographic distance. So we use a really simple, sorry for those in the back who can't see this is omega ij is the covariance between samples I and j. And we modeled it as just a simple exponential decay of this geographic distance dij between samples I and j. And the trick of the method is that we allow samples to sort of choose their own locations. So instead of being an observed quantity based on where you went out and sampled your organisms, whatever they are, we treat the parameter, or the sample locations as parameters that are estimated as part of the model, so that this dij is sort of an emergent property of those parameters. And then we can just calculate the likelihood of the sample covariance as Wishart with degrees of freedom equal to the number of loci across which the sample covariance is calculated. And we do our inference in a Bayesian inference framework, which is a custom Markov chain Monte Carlo. I'm not going to talk about the details of that in this talk, but I'll bore you to tears if you ask me about it later. So that was a lot, but I think it's sort of easily digested in an empirical example or simulated example. So here's a grid of populations. Each dot is a population that I simulated using MS. And there's nearest neighbor migration, and I'm just sampling every other population. So only these large colored blobs got sampled for the data set. And there's a barrier across the middle of this grid, across which the migration rate is attenuated by a factor of 5. So when we run SpaceMix, this is the result that we get. And this hopefully is really intuitive, right? What's happened is that each side of the map has sort of boogied away from the other side in a way that reflects this greater effective separation between them on that barrier. And this is another way to look at it. This is true pairwise geographic distance between each of these sampling points. And I'm plotting allele frequency covariance on the y-axis against it. And samples in red are comparisons on the opposite side of the barrier. So you can see per unit separation, samples that are on opposite sides of the barrier have lower covariance due to this action of the barrier in the simulation. And then this is geogenetic distance on the x-axis on the other panel of the graph. And you can see that what's happened is the samples have partitioned themselves in such a way that you get this single decay of relatedness with distance, but distances have changed, right? That's what this geogenetic map is all about. Here's another example, just to sort of build your intuition. Here's a simulation in which each population in this column of the lattice has expanded in the very recent past into all the other populations in its row. So when we run SpaceMix on that, this is the output that we get. So again, we're recovering the overall lattice shape, but now each daughter population of that expansion event is clustering very closely with the parent, which reflects this very recent sort of shared drift that they all share due to the expansion. And this is sort of highlighting what happens or why SpaceMix has some advantages over PCA. Here's another simulation, which is just a linear simulation, again with nearest neighbor migration, and just populations on a line. And when you run PCA on the genetic data that are simulated using this scenario, you get this kind of funky U shape. And this type of U is characteristic of a pattern of isolation by distance in PCA. And the reason you get a U has to do with the way that PCA does its decomposition. And in contrast, this is the results that SpaceMix gives you. These are the 95% credible ellipses around sample locations in space. And you can see that there's like a little bit of wiggliness around the edges, as you expect, because there's no simulation, or there's no migration from their neighbors on either side, because they don't have any. But it's recovering a line in a much more pleasing way than PCA does. So we wanted to take this out for a spin and use that same human data set to do it. So here's, don't worry about reading the names, hopefully you can get the gist of it from the colors, but this is that sample of 95 human populations that were sampled as part of this data set from Garrett Halenthal's recent paper. And this is the SpaceMix output, so this is the map that you get when you run the analysis. And it's kind of cool, so it recaptures the geography really well. Here's sub-Saharan Africa and North Africa and the Middle East. Here's a big Eurasian schmear. And then these are the Americas, and these are Oceania. So that's nice that it's sort of recovering the overall geography that we know humans experienced in the past. There's also some cool things. So if we zoom in now just on Eurasia, you can see that even within sort of Western Eurasia and East Asian samples, the sample relative positions are recovering their actual relative geography. So here's Russia, Finland, Lithuania, sort of in the north with Norway, and then you grade down through the French, Spanish, and Basque, and then Sardinian and Cypriot, and then into these Middle Eastern samples, Druze and Syrian. So, and the same thing's happening in East Asia. So that was kind of comforting to see that we're recovering that local geography really well as well. And then there's also this interesting question that came up with these samples that are in the middle of these Eurasian groups. So these are the Uzbekistani, Hazara, and Uyghur, for those of you who can't read it in the back. And these are 3 human populations that have really interesting and well-studied histories and genetic histories. And the fact that they're falling intermediate raises this question about whether they are truly genetically intermediate in the sense that they are geographically between these two and therefore just belong there, or if they're admixed actually between these two East Asian and Western Eurasian sample sets. So by admixture, I mean that I'm referring to a scenario in which there's 2 or more parental populations, each of which are contributing to the ancestor of some sample that you get to see in the present. And the extent of that contribution are given by these weights. So this is the weight that sample C gets from parental population A, and same with B. And another way to think about them is the probability that an allele that you see in C came from A or from B. So what happens to SpaceMix if there's admixture in the scenario? So here, this is the sort of same basic lattice, but now it's kind of like Clue, and there's a secret passage between the corners, and population 30 looks half like itself and half like population 1. So this is the result that SpaceMix gives for that scenario. And it's a little higgledy-piggledy, but it also makes sense in the sense that 30, which is the truly admixed population, is kind of folding space around itself. It's correctly choosing its nearest neighbors, right? 29, 28, 25, 1, 2, and 6. But because it's intermediate between these two sort of groups of samples, because it's admixed, it has to bend space to describe itself. Which is cool, but also not particularly intuitive, especially when you don't know what the true simulation history was. So we wanted to explicitly accommodate admixture in the model. And the way we did that is by giving each sample a floating point in space from which it draws admixture. And both the location of that point and the amount of admixture it draws are estimated altogether as parameters in the single model. So here, for example, this is the location that sample J has, and it's drawing admixture from J star, and doing so in amount Wj. And this is, again, the probability that an allele that you see in J prime, or in J, was drawn from this location in space at J star. So another way of saying it is that just the allele frequencies in sample J are a linear combination of the allele frequencies at this location it chooses, J prime, and the location of its source of admixture, J star. So if we have now 2 samples to sort of increase the complexity of this cartoon, there's J and I, and each of them are drawing admixture from a different point in space, J star and I star, and doing so in different amounts, Wj and Wi. And here the thickness of this arrow represents how much admixture J prime is getting from its source of admixture. And then we can create this admixed parametric covariance matrix by just walking through all of these combinations. So here, this omega di prime j prime is the geogenetic distance between samples I and j, and it's weighted by the probability that an allele you see in either did not come from its source of admixture, which is 1 minus wi and 1 minus wj. And then you can use the same logic of weighting the spatial covariance by the probabilities of where alleles came from to work through all of these other terms and end up with this overall admixed covariance. And then we can proceed with inference as before, so this was the original model, and now we just add prior probability distributions on the sources of admixture and how much admixture you're drawing, and do the same inference. So this was the original simulation scenario. Here was that weird, like, inverted vacuole graph. And then this is the result when you run it with admixture. So here, each sample gets to choose admixture from any point in space and any amount of admixture between 0 and a half. If you want more than a half, then you could have switcheroos between the source of admixture and the actual sample. And I'm denoting the strength of admixture by the opacity and width of this arrow. And as you can see, only one admixture arrow shows up, and it's from population 1 directly to population 30. So that was a really nice indication that we can actually estimate admixture pretty well. And again, this highlights another problem that PCA has. Here's the PCA map that you get for that same simulation scenario. And you can see that sample 30 just falls in the middle. So it does this thing where it looks half like itself and half like 30, and so it just puts itself right in the middle, which makes sense, except that you can't disentangle that possibility that it's admixed from the possibility that it was actually just a sample that belongs right here, that's true geographic, or sort of, genetic flavor is as if it came from between 13 and 18. And that's been addressed in a number of other papers recently from John November's group. So what does the human map look like with admixture? This is what it looks like with admixture. So this is a lot. There's a lot of information here. And I'm going to walk you through it very briefly. So each sample's name shows up twice. Once in bold, which is the location that it chooses for itself, and once in italics, which is the location of its source of admixture. And again, the arrows, the width and the opacity of the arrow is proportional to how much admixture samples are drawing. So here's just highlighting one example. Here's the Ethiopian sample that are choosing a geogenetic location down near the sub-Saharan African cluster, but drawing a lot of admixture from a location near the Middle East and sort of southwestern Eurasia. So overall, we're again recovering the geography of what we know about human expansions and human origins really well. Here's this long sort of distance between the Eurasian, American, and Oceanian samples and the sub-Saharan African samples is really strong indication of the shared out-of-Africa drift that all of the non-sub-Saharan African samples shared. But you can also see that there's been a lot of subsequent gene flow, especially between North and Sub-Saharan Africa, and that's these admixture arrows here. And then we can zoom in just on the Eurasian samples again and look at those three kind of interesting populations from the first analysis, the Uyghur, Uzbekistani, and Hazara. And you can see that, for example, the Uyghur are choosing a location that's close to these Eurasian samples, but drawing admixture in a large amount from a location that's close to these East Asian samples. So they do seem to in fact be admixed and not sort of geographically intermediate. And for fun, so the Coop Lab all got our 23andMe done. And so for fun, we sort of placed ourselves marginally on this map. So that's what this looks like. This is me. This is Graham, who's British, for those of you who don't know Graham, and kind of boring, he shows up with the British. My ancestry is largely Ashkenazi, and there's no Jews in this extant sample that we used. So I choose this location that's European, but I'm drawing non-trivial admixture from a location that's off sort of towards North Africa, which is what a lot of the other Middle Eastern samples do. And then the most interesting one is Elisa. Elisa, who just is filing her PhD work today, which is really exciting. Elisa's parents, one is Japanese, one is Iranian, and she's taking a location that's right on the Japanese sample and drawing admixture directly from the Iranian sample, which was both a nice confirmation that the method works and also Elisa's parentage. So that's SpaceMix. That was kind of a blitz tour of it. I'm happy to talk about it more. But just to sum up, patterns of genetic variation are very often complicated. They're shaped by a lot of processes. And it's really helpful to be able to visualize them in sort of an intuitive, two-dimensional summary that captures a lot of information about the data. So that's what SpaceMix is designed to do with these geogenetic maps. The next next project that I'm talking about, which I'm working on currently, is a method that I've been working on to disentangle or to simultaneously study continuous and discrete patterns of genetic differentiation. So there's a lot of examples of discrete population differentiation when we look in nature. One of the canonical ones are these greenish warblers, which have these different morphs that are distributed. And there's strong patterns of discrete differentiation between them. And then if we look in phylloscopic Stropholoides, you can see that there's also really strong examples of continuous gradations in sort of genetic flavor that are also geographically dispersed. And as I'm sure everyone knows, these are in fact the same system, right? Greenish warblers are phylloscopic Stropholoides. And this is the famous ring species from Darren Irwin's research group and others, Ticehurst, that's distributed around the Tibetan Plateau. And this is kind of a cartoon in some ways that I'm showing now, but it's also, I think, something that's indicative of a lot of natural systems, that there is both continuous differentiation across space or across time, and also discrete patterns of structure across barriers that may be phenological or behavioral or physical, like a mountain range or a river. And this is sort of one of the classic problems in the inference of population structure. What do you do when you have both of these patterns? And it's called sort of the Kleins versus clusters thing, but if you have a continuous sort of Klein of genetic flavor, which you sample discretely, then it's often very difficult to figure out whether there is in fact continuous structure across all these samples or a discrete break between them. This is partly an issue of just sampling efforts. It's hard to sample uniformly and continuously across an entire range of a species or population. But it also deals with the fundamental failing of our population genetic models that they can't accommodate simultaneously discrete and continuous structure. So we're calling the method that we've used, or that we've created to deal with this geostructure, working title. And basically the way it works is, well, I'm going to talk about the way it works and with an empirical example, which again is not a vertebrate, sorry. And I'm going to walk through sort of why this is important in a lot of systems, how this works, and then give you some quick results. So the system I'm using is populus in the Pacific Northwest. So these are two different species, Trichocarpa and Balsamifera. And Trichocarpa will appear in green throughout the rest of the talk, and Balsamifera in black. And this is the range. These are 35 drainages that were sampled as part of a recent paper from Armando Herraldes. And yeah, so Trichocarpa is sort of all along the coast here, and Balsamifera is more sparsely sampled in the interior. When Herraldes and al. Ran structure, they get two clusters, as you'd expect, that conform. This is lumping all the Balsamifera together. They get two clusters which conform, as you'd expect, to this species structure, with some admixture in these geographically intermediate populations of Trichocarpa. And then when they look specifically within Trichocarpa, running admixture, they get, I think, a best K of 7. So there's a lot of differentiation within Trichocarpa, but interestingly, you see a really strong indication of isolation by distance, right? So there's strong spatial autocorrelation in cluster membership along this swath of sampling. And this is sort of one of the classic telltales of isolation by distance being captured by this discrete model of structure or admixture. So looking at that just at a single locus, this is real data from this data set of about 33,000 SNPs. So here I've just plotted the allele frequencies in each drainage at this locus, the black bar at the mean. And now this is plotting each of the drainages with the same heat color. So hotter colors are higher allele frequencies. And hopefully two patterns jump out pretty clearly. The first is that there's discrete population structure. So across this very small geographic separation, there's a relatively large difference in allele frequency at the single locus between Balsamifera and Trichocarpa samples. But also there's clearly continuous differentiation, especially within Trichocarpa. These two samples that are close together have very, very similar allele frequencies, and more similar than a comparison between Trichocarpa that's between a pair of samples that's farther away from each other. So we can take these two samples that are close together, and you can see just on this single locus they have very similar allele frequencies, and we can describe this, this tendency to co-deviate in the deviation from this mean at each locus as the sample covariance quantity, which is given here. This is just the sample covariance between samples I and j is given by this shared deviation away from the mean across all loci, divided by the number of loci that you have. And we can calculate this for all the loci in the data set and get a really fine measure of how these samples are related to each other at a whole genome scale. So that's what this quantity looks like. This is the sample covariance on the y-axis plotted against geographic distance, and again colored with sample A and sample B as the inner and outer colors. And some cool things pop out. So these green on green comparisons are Trichocarpa. The black on black are Balsamifera, and then the green and black are the comparisons between species. And you can see that for the majority of these between species comparisons, there's much lower allele frequency covariance per unit geographic separation than either of the within sample comparisons. So this is an example of sort of a strong indication of there being a barrier of some kind between these. But also, right, you can see there's a clear indication of this continuous decay of relatedness, especially within Trichocarpa, where covariance decays with the separation between them. All right, so that's the empirical example. And the way that geostructure works is that we model discrete population clusters within which there's some continuous decay of relatedness with distance. So this is a cartoon of that. Here are trees in each of two different clusters, right? And within each of these, we have some parametric model of how covariance decays with distance. And here, these are color-coded, and you can see that the shape that they're taking is different between them, so they're not constrained to be the same. And we call these spatial functions of covariance f indexed by the cluster k from which this curve belongs. And this is just describing something where, for example, these two trees, which are closer together in this cluster, would have a higher covariance than this pair of trees that are farther away. But the rate at which this covariance decays is heterogeneous between different clusters. And then, like structure, we allow individuals to be admixed between these different discrete groups. And the admixture proportion we denote by these, again, by these W's, these weights, which are indexed by the cluster to which each of these, from which each of these samples is drawing admixture. And then we can calculate, in a very similar way, this parametric covariance. So just walking through this briefly, this is the cluster-specific spatial covariance. This is a parameter that governs cluster-specific shared drift. So for example, if a population has a historically small effective population size, they'll have a larger deviation away from some global mean allele frequency at each locus. And we can describe that differently across different populations. And then these are the admixture proportions, just summed across all of our K clusters. And we can take the likelihood of this parametric covariance function again as Wishart, which is nice and efficient. Yeah, so returning to our empirical data set, what does this look like? What do results for this look like? So k equals 1 is not that interesting, right? They're all drawing 100% membership in that one cluster that exists. But this is a plot of the sample covariance in black with the parametric covariance plotted on top of that. And you can see that the fit's not great, right? It's not doing a great job with k=1, which is not surprising because we know that sort of biologically there should be at least 2 clusters. But what's cool is that even with one cluster, we're capturing something spatial about the data, right? The relatedness is decaying. These red points show the decay of relatedness with distance in the parametric estimate. When we go to k=2, we see that there's sort of strong concordance between this model and the structure analysis that Heraldes et al. Ran. But what's neat now is this is plotting the 2 different spatial covariance curves. And you can see that one is corresponding mostly to the balsamifera comparisons, and one mostly to the trichocarpa comparisons. And just going back real fast, you can see that, again, these trichocarpa samples are almost all in blue, and the balsamifera are mostly in red with some admixture in between. So we can plot, again, model fit as before, and see that it's really good, right? That there's clearly some outliers, but in general, with only 2 clusters, we're able to really strongly capture whatever, or describe whatever process actually generated these patterns of differentiation. So that's sort of a really exciting finding. And in some ways, it's not surprising because we have more parameters than a sort of vanilla structure model. But it's also nice because we're describing something that's biologically real. We think that each of these different clusters is just, potentially experiencing continuous differentiation in space, and the only discrete barrier is between these 2 different subspecies or species. We can also do model comparisons. Actually, in the interest of time, I'm going to skip this, but we can do model comparisons, and we see that in general we want less k, which is nice. And then there's some brief extensions that we're looking at now are looking at relationships between clusters. So right now we treat each cluster from a structure analysis or in geostructure as if they sort of were fully formed and unrelated, but that's obviously not true. And we can describe the relatedness between different clusters using a tree-like scenario where, for example, clusters green and blue are more closely related to each other than either is to red. Or spatial scenarios where, for example, one cluster, the red cluster, was derived from a point in space within this blue cluster at some point in the past. And this is a really nice model for describing things like parapatric speciation, which maybe happens a lot. And the other thing that we're excited about is extending this to temporal relations. So right now, this spatial covariance is just a function of the geographic distance within each cluster between samples. But we can also make it a function of the time between samples. And as we get more and more historical specimens that are genotyped, this is maybe a really useful tool. So this is kind of a difficult to read plot of humans. This is a preliminary analysis of humans in Western Eurasia and Northern Africa. And I don't know if you can see it. Yeah, so here's the side sample showing sampling back in time over the last 5,000 years. And we can estimate admixture proportions. This is with k=3. So these analyses are still going, but I'm pretty excited about where this can go. And then the last thing I'm going to talk about, just blitz through, is the project that I'm hoping to work on this year with Bree and Michael. So this is looking at genomic consequences of a changing environment. So obviously we're all really interested in local adaptation and climate change and gene flow. There's a lot of things, especially out of Sally Aitken's lab, and then a lot out of the MVC, Emily Rubage's paper, and Kevin Rowe, Rappucciola. And there's simultaneously just advances in DNA sequencing technology are making it easy for us, or at least doable for us, to genotype historical specimens, either from museum collections or archaeological digs. And this is really exciting that we can link these two together now to answer questions about both. So one thing that's cool about historical or ancient DNA is that we can use it to polarize the direction of gene flow, right? If we have 2 samples in the modern day, it's hard to figure out how they're related. We just know there's a single quantity that describes differentiation between them. But if one is sampled in the past, we can use it like a fossil calibration on allele frequencies at a point in space at a point in time. And then we can learn about how allele frequencies have changed at that point in space through time with a change in climate. So this is kind of the cartoon of how I think about this. Here's a point in the past at time 1, and there's some environment that's heterogeneous over space at that point in time. And then through time, the environment is changing. So you can see if we follow this point in space up, the environment at that point in space has sort of shifted, and this is kind of a cartoonish shifted north to higher latitudes. And we can ask questions about how populations from the past and the present are related to each other across this shifting environment. And specifically, imagine we have 3 points in space, A, B, and C, sampled at time points 1 and 2. We might expect that individuals who disperse from, let's say, B, following the direction of the changing climate, might have a higher fitness because they're sort of staying in this envelope to which they're locally adapted. And then we can ask questions, given some rate of decay of relatedness with distance, with distance and with time, we can ask whether, for example, B1 is more closely related to A2 than it is to B2 or to C2, even though the geographic separation between and temporal separation between A and B and C are the same. There's this sort of other factor of the changing climate that we can try to look at. This slide is maybe slow to load. Yeah. So this is the type of thing I'm talking about. So here's a comparison in blue. So if we imagine that the direction of climate shift is sort of northward at a point in space, so whatever flavor of environment was at a point in space at some point in the past, it now looks more like a point south to it in the present. And we can try to model this effect where comparisons in this allele frequency covariance between samples, given some rate of decay with distance and with time. There's a lot of really exciting work on both of those coming out of Berkeley and also Nick Martin's lab. We can ask whether we can see some effect of climate change in structuring whole genome patterns of relatedness across space and time in this changing climate. So I've been proposing to do this with Paramiscus from the MVZ. So this is a map of Paramiscus samples across a map of the states that's heat mapped by the time for which they're sampled. There's a lot of specimens in the collections here. And I think it's a really exciting opportunity, especially because we know a lot about changing climate in the West, to answer questions about, for example, how selection is happening, and whether or not we see a signature of selection at different points along the genome in the species. And also thinking about sort of shifting mosaics of coevolution with other species in the same communities, and I'm excited about extending this too. So these are the 3 projects I've talked about. I'll be around and sort of split between Michael and Bree's lab, and I'd love to talk to you more if you have any questions. And I just want to reiterate that these are sort of the major questions in my research program, and I want to thank all of you for listening, and especially Michael and Bree and their labs have been super warm and welcoming since I got here. And I just want to say, lastly, that I'll be starting a position at Michigan State as assistant faculty starting in the fall of 2016, and I'm looking to recruit especially postdocs, but motivated students as well. So if you're interested in that, or if you know someone who's interested, please contact me. And with that, I'll take questions.
So you mentioned selection at the very end.
Yeah.
But you did mention it through the rest of all of the talk. So I'm just curious, when you have 30,000 markers, presumably over large geographic scales, presumably a bunch of them are under selection. So I'm just wondering how you figure selection into the patterns and how that influences the inferences derived therefrom.
Yeah, that's an interesting question. I think a lot depends. So you said at the beginning a lot of those loci are presumably under selection. I think
Some are in.
Sure. I think that the extent to which selection is going to be shaping whole genome patterns of relatedness between samples is a function of the strength of selection, obviously, and also the spatial distribution on the genome of loci that are under selection. So for hugely polygenic traits that are under strong local adaptation, so migrants will be really selected strongly against, there might be some whole genome signature of divergence, right? But if that's not the case, it's very likely that although there'll be some small uptick in divergence genome-wide between a pair of samples that, or a pair of populations across which migration is selected against, you may actually not see that strong a signature because it'll only show up in these introgressed, or regions that are not introgressing.
So that may be true or it might not be true, but the other possibility is that the informative markers are the ones that are under selection. So although there's relatively few of them, they're the ones that are showing important spatial patterns, which would then, the model would then use to make the inferences about space.
Yeah, I mean, I guess it depends on what theta in your sample is. And we can talk about more of this afterwards, but I guess I would say that, at least in my worldview, a lot of diversity is neutral, and it's linked potentially to things that are under selection. But I guess I would be surprised if the majority of the signature there is being driven by selection as opposed to, or directly by selection, as opposed to potentially migration just being limited between genes and then drift acting locally. Yeah, but it'd be fun to talk more. Yeah.
When you talk about the time calibration, how long do you need to set the point? Like, could it be a museum specimen within the last 50 years, or are you actually talking about like a fossil specimen?
Yeah, I mean, I think more the former than the latter. So a lot of these models of looking at allele frequency changes sort of compounded across the whole genome to quantify genetic differentiation break down over really long timescales, basically because drift is bounded right at 0 and 1. And so if a long enough time passes, you're really unlikely, you're likely to underestimate the amount of differentiation that's accrued because loci will become fixed and they can't become more fixed. So in the Peromyscus project, I'm thinking over the last 50 to 150 years. Yeah.
I was going to ask about, so in the Peromyscus sampling, it looks like there's pretty good sampling over that whole period,
Yeah.
except I noticed in the early 80s nobody was out collecting for some
Yeah.
reason. But there's pretty good sampling, and so do you think that having time series data with lots of points adds a lot of power for making inference compared to having a couple of points that are really well sampled in time but with long intervening intervals? And I mean, just to provide a little context, there were big survey efforts by Grinnell and others at particular points in time, and some of those have been repeated, and people in the museum are about to start another repeat of that. And so these are places that are going to be very well sampled at particular points in time, but potentially sporadically, or not with this sort of continuous distribution.
Yeah, so this is something I've been thinking about a lot as I try to figure out which samples would be best to target with a non-infinite budget. And I think that some of it depends on, so it depends on what you're looking for. If you're looking for the signature of selection, so alleles that you think are locally adapted to a particular set of environmental conditions that have shifted over space, then if you've sampled well at a single point in space, and so in the limit, a very small point in space, at multiple time points, but not broadly in any given slice of time, you may miss that because the environmental envelope may have moved faster than your sampling efforts. And so you'll see things that are different, but it'll be hard to correlate what's different with the potential causal mechanism of local adaptation in shifting environment. And likewise, I think it depends on the scale of dispersal. So it may be that these patterns that I'm thinking about of higher covariance between a point in the past and a point in the present that have more similar environments, even if they're farther away spatially, that those are playing out over tens of meters instead of many kilometers. And in that case, if you've sampled either too broadly spatially or potentially temporally, depending on the rate at which you see climate change moving environments across the landscape, you might miss that. So I think the first pass is to think about areas of the landscape in which environment has been changing the most over your sampling period, and then use those to focus your efforts. Because it's sort of you're most likely to pick up something cool. Yeah.
I mean, in practice, there's often a trade-off, right? And so, I mean, you could sample broadly geographically, and sampling over multiple points in time almost always requires multiple investigators living it. I mean, it depends what the time scale is that we're talking about. But.
Yeah. I mean, I think the ideal would be, 100 samples per cubic square mile year, right? And that's just not, it's not possible. So I think, I think that's not something that's likely to happen in the near future. Yeah.
Other questions? All right, well, at least on Wednesdays and Thursdays, Gideon is in the office next to mine, and early in the week, you're over in.
Yeah, in 147 Berkeley.
Okay, thank you very